Good morning, good afternoon, good evening, everyone, and welcome to this Inside Scientific webinar titled Vascular Tubular Crosstalk in the Renin Angiotensin System. My name is Darius Sulam from the events team here at scientist.com, and I'm very pleased to be your host today. This webinar has been sponsored by Alzet Osmotic Pumps and produced in partnership with the American Physiological Society. So a big thanks to them for helping to produce the webinar and making it possible for us to share science together. We're very fortunate to have Dr. Matthew Sparks, who will be discussing his recent and exciting new findings on the vascular to epithelial system in the kidney that contributes to blood pressure control. I'd now like to welcome Dr. Matt Sparks to the stage. Hello, Matt. Thanks for joining us today.
Well, thanks for having me.
The floor is yours whenever you're ready.
Okay. Well, thank you so much for inviting me. And this is definitely a topic that I enjoy discussing. My topic will be about vascular tubular crosstalk in the Renin Angiotensin system. I don't have any conflicts of interest related to this talk other than a small stipend I'll receive for doing this presentation. The background about the Renin Angiotensin system will be what I'll first talk about, and then I'll talk a little bit about what's known about the vascular action using experimental models. And then I'll discuss some recent findings that we had in our research. This is the Brazilian pit viper, in which the very first compound, captopril, that blocks the Renin Angiotensin system was first identified from. And I like to go back to the original publication back from 1971 from Bothrops Jakarta. Shortly thereafter, in 1974, this paper in New England Journal of Medicine was published, in which the medication captopril, at that time called SQ14225, was given to human subjects. And as you can see, in two separate conditions, in 6 patients each, essential hypertension, that's what it was labeled back then, and renal vascular hypertension, you see a large drop in blood pressure by about 30 millimeters of mercury. This was also seen with a large increase in plasma renin activity, which is something you see today. And then we also see really a diminished in the angiotensin converting enzyme, key pathway that's blocked with captopril. So many studies have been conducted in humans since that time and showed reductions in cardiovascular disease, reductions in kidney failure. This is one such study called Renal, which has studied the ARB losartan in patients with type 2 diabetes. You can see the composite outcomes were greatly reduced in patients using these agents, almost 6 or 7% reduction in end-stage kidney disease, and also in the primary composite outcome, a much better outcome as well. So these are now standard of care. This is another paper looking at its use in heart failure. You can see just a dramatic decrease with the use of enalapril with patients with congestive heart failure. As I mentioned, this is standard of care now, and we're now layering other therapies on top of this, including SGLT2 inhibitors. But we really are still trying to unravel how this system works, and our laboratory has really spent the last decade understanding how the vascular effects of the renin-angiotensin system affects blood pressure. As we know, activation of this system is critical in the pathogenesis of both cardiovascular and kidney disease, both linked by high blood pressure. Blockade of this system reduces morbidity and mortality. And we also know that prevention of cardiovascular events is partly independent of blood pressure. And these are studies where you give patients 2 classes of drugs. One includes RAS inhibition, the other one includes other classes. And you see greater results in outcomes with a similar reduction in blood pressure with patients on RAS blockade. Because of these things, it's really important to understand the molecular action so you can exploit the positive ones and also try to diminish the negative ones. We do know there are significant side effects of these medications, including cough, including hyperkalemia, and occasionally acute kidney injury. As we have uncovered more and more actions of this system, we get more and more complicated. One of the central regulators, though, is angiotensin 2 and its action on AT1 receptors. Most of the actions of AT1A receptor are considered deleterious. However, we have seen, for instance, in the collecting duct, intercalated cells, and sometimes in immune cells, that these actually can work to dampen the hypertensive response. Our laboratory has focused on the AT1 receptor in removing it from different cell lineages. One of the challenges about understanding this system is that type 1 angiotensin receptors are located on basically every cell lineage in the body. And importantly, they're located at all the cardiovascular centers and tissues that we have. And I'm going to sort of focus more on the vascular system, which is sort of traverses all the systems, including the kidney, the brain, and talk about what is known about its actions. The very first knockout mouse was published by my mentor, Dr. Kaufman, in 1995. And if you had a global knockout mouse, you have a very large decrease in blood pressure by almost 40 millimeters of mercury. And even a heterozygous knockout mouse had a phenotype. This indicates that just in the basal state, this receptor is really important for the maintenance of just resting blood pressure. But what about its vascular action? So if you infuse angiotensin into these knockout mice, what happens? And if you see on the far left, in the wild type scenario, this is just infusion of these of angiotensin 2 into the mouse. You have a big increase in blood pressure. These are acute responses. These are not chronic responses. And then it comes back down to baseline. In the heterozygous knockout mouse, you have a blunted effect, but it's still a pretty decent effect. However, in the global knockout, you just have a hardly any response at all to angiotensin 2. It is likely that that small response that you see is from the AT1B receptor. Rodents, mice, and rats both have an AT1A and AT1B receptor, whereas humans just have one isoform called the AT1 receptor. The AT1A knockout mouse, since its phenotype is so profound in both the basal state and in infusing acute angiotensin 2, is considered the human or the mouse analog of the human AT1A receptor. And thus, we focus most of our studies on the AT1A knockout mouse. As we move forward in time, Steve Crowley, who at that time was a fellow in the lab, worked with Tom Kaufman to develop this mouse model where we sort of try to understand what tissue compartments are really responsible for this. And so he used a kidney cross-transplantation model. In this model, they take out both of the kidneys and then replace, by kidney transplantation, another kidney, even in the wild type state. So if you look here, this is the wild type mouse that has a wild type kidney put into it to serve as a control. And you can see the blood pressure. This is the basal state. These mice all have radiotelemetry catheters implanted into them, and they're allowed to sort of recover for a few weeks after this subsequent surgeries. They have the bilateral nephrectomy, and then they have a single transplantation that's put in. If you have a scenario where the whole mouse, all the tissues in the body have the angiotensin receptor except for the kidney, the blood pressure is reduced by about 20 millimeters of mercury. And similarly, if you have the opposite scenario where the entire mouse lacks angiotensin receptors, but only the kidney has them, you have a reduction in basal blood pressure by 20 millimeters of mercury. And if you have an entire knockout mouse, you have the blood pressure that's all the way down. And so this is a really important study that demonstrated to us that both systemic and kidney angiotensin receptors are both contributing to basal blood pressure. And for my research moving forward, it really became important to understand that the vasculature, which is in both those compartments, could be playing a role. Now, what about the infusion of angiotensin 2? And so in this experiment, the exact same mice that I had previously described and we had their basal blood pressure measured. Now we infuse angiotensin 2 via osmotic mini pump, about 1,000 nanograms per kilogram per minute. That's our standard dose that we use in our laboratory. And if you look at this experiment, we wanted to see, all right, well, what happens in experimental hypertension? And if you look right here, this path here is what you see in the kidney knockout mice. You see that the blood pressure goes up and then sort of comes down. The very bottom one is the global knockout mouse. You really don't have any response at all. And the wild type control is the top one. And you see a big response to angiotensin 2 that's sustained over the infusion period. Now, the systemic knockout has a blunted response, but towards the end achieves a blood pressure like the wild type. So the blunted response is only in the early stages of hypertension development. So this experiment was really important in really establishing the primacy of the kidney in the maintenance of hypertension. But also, it did show a contribution of the systemic tissues not quite as profound as the kidney. So this really set the stage for really the next decade and a half or so of research and trying to explore this system further. It also was the exact time that Cree-LoxP technology was becoming widely used, and I joined the lab shortly thereafter in 2008 and really started to explore the vascular system, whereas Steve Crowley focused more on the immune system, and then Susan Gurley focused on the nephron. So the two clinical questions that I wanted to answer was, one, how do vascular angiotensin receptors control blood pressure? And two, what is the contribution of the kidney versus systemic vascular angiotensin receptor regulation? In order to answer this question, we used mouse knockout technology using Cree-LoxP. And we had several different conditional knockouts. And I think one of the challenging parts of this research is that not every Cree mouse line will function the way you think it functions. So it's really important when you get these mouse models to figure out, like, where is it expressing Cree? How much is it? You know, how big of a knockout do you get? Are there any other tissues that are expressing Cree recombinase? So we had made the AT1A knockout floxed animal, so where you have a flox allele placed, so that Cree, when it recognizes it, can delete the gene. And then we crossed them with that second mouse line. And we use the SM22 knock-in promoter, and that's important because that's a very faithful expression model whenever you knock in a Cree into the endogenous promoter. Another technique is where you make a completely heterologous transgene that's just randomly inserted, usually multiple copies in a random part of the gene, which can do two things. One, it can knock out some random gene, or two, it can sometimes it's hard for the chromatin structure to unfold in the way to open up that structure, so it doesn't have as good faithful expression. SM22 is also called transgalin or taglin. And this is sort of a description of the mouse that has the Cree that's driven by a promoter, in our case, the smooth muscle 22 or SM22 promoter, and our target gene, which is in our case was the angiotensin type 1 receptor, which has these LOXP sites. These are sites that are specifically recognized by Cree recombinase to delete the gene. And so after you do two successive crosses, only in those smooth muscle cells or whatever cell type expresses SM22, you lose the gene. This will allow us to study very similarly to what Steve did, but instead of having just a complete organ transplant, we can now remove it just in specific tissues. The next step is to look at faithful expression, and we do that with a reporter mouse. This is called the MTMG, and an MTMG mouse has every cell in the body expresses membrane tomato, which fluoresces red without the use of any other substance. You just basically take the tissue out, look at it under the microscope, then it will fluoresce red. And then if Cree recombinase is present, it turns green. So it turns off red and turns on green. And so if you look at it, you can really get a good sense of where this expression has a very faithful expression in vascular smooth muscle. Now, we have a prior publication where we had the heterologous transgene SM22 mouse, and that does not hit some of the smaller vessels and really is restricted to large conduit vessels. And for these studies, I'll talk about the knock-in of the KI SM22 Cree mouse. These are just more examples of this is the inner medulla. This is, again, inside the cortex. You can see all the vessels here, and then this is sort of a blown-out view of the kidney. So you can see lack of expression in the tubules, but you have plenty of expression all throughout the vascular structures. The next step is see, all right, well, do we knock out our receptor? And so in this, we have liver. There's no change. This is mRNA. There is not a good commercial antibody that can detect the angiotensin receptor, just like many GPCRs. And you see there's no deletion in the liver, no deletion in adrenal gland, no deletion in brainstem. You do have massive reduction in mesenteric vessels and also in the aorta. So after this data, we're saying, all right, we've got something that looks like has good Cree expression in our mouse system, and we've knocked out the receptor in which we're trying to. The next step was to look at the blood pressure phenotype. And so we then implanted telemetry catheters into the carotid artery. And these are non-anesthetized animals after they've recovered for 1 week. We look at their blood pressure over 1 week average, and you see there is a reduction in blood pressure by about 10 millimeters of mercury. I will also say that these are all the data I'm presenting here are male mice. We have done all the experiments on female mice, and that's all published in AJP if you want to check that out as well. The next step is to look at salt sensitivity. And one of the phenotypes that we saw with the angiotensin receptor global knockout mouse is that if you give them salt back, their blood pressure will correct, because a lot of the abnormalities for the angiotensin receptor is to have more urinary loss of sodium. And so the other thing to note is these are all in a black 6 background, which are very, they do not have salt sensitivity. So if you give them large amounts of salt, their blood pressure stays the same. And you can look on the top side with low salt, and really this should be no salt because very, very low salt diet. Blood pressure barely changes. And then you give them high salt, doesn't really change much either. However, in the smooth muscle knockout, you do have a drop in blood pressure with low salt, meaning that they were really dependent on any of the salt they had in their regular diet just to maintain that blood pressure. And then if you give them a high salt, you really have an increase in blood pressure. So basically this unmasked the phenotype of salt sensitivity and also shows that there is likely some abnormality in sodium homeostasis that is occurring that we'll explore further on. The next step in our studies is to look at hypertension pathogenesis. And again, we use the chronic angiotensin 2 infusion using the osmotic mini pumps from Alzette. And this is 1,000 nanograms per kilogram per minute. This is a standard dose that we've used throughout all of our experiments over the last several decades in the lab. And you can see just from the baseline data, they have a blood pressure difference, as we mentioned earlier. And then you have a rise in blood pressure in the controls that kind of flattens out over time. However, in the smooth muscle knockout of the angiotensin receptor, this response is blunted throughout the entire infusion period and is reduced by almost 40%. So this shows two things. One, that vascular smooth muscle angiotensin 2 receptors are responsible for the maintenance of chronic blood pressure, but also participates in the generation of the hypertensive response to angiotensin 2. Now, these findings weren't necessarily surprising. I will say that we found a very similar phenotype in the proximal tubule from Susan Gurley's work. And so if we really take that into context with this, it seems that smooth muscle and proximal tubule are two of the main tissue compartments driving hypertension in the renin-angiotensin system. But we really wanted to go deeper in here. And so also I mentioned earlier the female mice, and this is the data from the female mice that we published a few years ago that show a very similar phenotype. The next step in our studies is to really look at the microvasculature of the kidney, and this is our laboratory at the Durham VA where we do all of our physiologic studies on our mice. And if you can see in the very back is all the blood pressure tracings, and we have the ability to do laser tissue Doppler. We have small ultrasound probes, and even the ability to do continuous oxygen consumption. So this is an experiment where we look at the systemic blood pressure response, and we put a blood pressure monitor in the carotid artery, and then in the internal jugular vein, we infuse boluses of angiotensin II. And so the first 3 humps here are increasing doses of angiotensin II, which are done every 10 minutes. And then at the very end, you can see a large dose of epinephrine. The control mice are in black here. You see a large increase in blood pressure as you get increasing doses of angiotensin II. However, in the smooth muscle knockout mouse, you do have a fairly blunted response, and there's really no difference in epinephrine. What struck us about this, though, is that the response, although blunted, was not absent. And so that really we question, like, what is the reason for that? Because if you really were to believe that the type 1 angiotensin receptor is very, it's kind of the main effects, it's called angiotensin, blood pressure constricting. What is the reason for this? And we saw the global knockout mouse and sort of what happened to it. So what is the mechanism? And so here's sort of what we're thinking here is one is the persistence of AT1B receptors, and maybe even upregulation of those receptors could be responsible. Could it be a stimulation of the sympathetic nervous system? And that's what I'll sort of talk about a little bit more, and we'll have some experiments to sort of define that. And there have been some work with the global knockout mouse to show that they have an enhanced sympathetic nervous system, that these two systems are linked. So we basically brought in another mouse line, and this was it's a smooth muscle myosin heavy chain. And this basically is tamoxifen sensitive Cree system. So you can knock out any gene in the adult life, or really any time you want to, by giving tamoxifen 5 days in a row. And so we had 4 different mice that we created. We also bred these with the AT1B knockout. So this is really a challenging model to build up. And so I'll kind of talk you through it. So the top one, basically we don't give tamoxifen to, and we have the AT1B receptor that's just wild type. And we don't, so it's not a knockout, it's just a control, okay? The second one is a control in regards to the AT1A receptor, but a knockout in regards to the AT1B receptor. So I would say this is control 1B. You see, like, what kind of blood pressure response does that mouse have to acute angiotensin II? The SMKO is the inducible smooth muscle knockout. And we have 2 flavors, one with the AT1B receptor and one without. So this allows us to sort of answer several questions, and one of which I didn't even pose, and that is there some alteration in a mouse that had a constitutive knockout, like in the other SMKOs, or is there a difference when you just knock it out de novo as an adult? And so let's go ahead and just put those through our experimental protocol. And the first is our acute vasoconstrictor protocol. And so these are the mice, the control, the 1B knockout control, the inducible with 1B, inducible without. And you can see with angiotensin II, there's really no difference between control and AT1B receptor. And again, showing the AT1A receptor is really what's responsible for this blood pressure response and not the 1B. The next question was about the inducible with and without the 1B. And you see there really was no difference. You had that blunted response, but that did not really change by the removal of AT1B receptor. So that really started thinking, all right, is there something else we're missing here? And so what we did is we gave the antiadrenergic blocker of phentolamine to block any response that you might get from the sympathetic nervous system. And as you can see, when we did that, we had really almost complete absence of any vasoconstrictor response in the mice that we gave that to, indicating that there likely was an enhanced response of the sympathetic nervous system. And this response is likely mediated by angiotensin II receptors in the brain, because the global knockout mice do not have this response. Now, do we have evidence of the sympathetic nervous system being overactive in these animals? And so we just measured urinary norepinephrine content in mice at baseline and after 3 days of angiotensin 2. And you can see even at baseline, these mice have an increase in norepinephrine content in their urine, and after 3 days of Ang2. And these findings were actually corroborated by another group that has the global angiotensin 2 receptor knockout mice. Just to see about what's, is this adrenal gland overactivity, and does not appear to be the case. We looked at all the different constituents that make norepinephrine from the adrenal gland, and there's really no difference. I just put a warning here because the next slide I'm going to show some animal studies, and this is our acute studies in mice where we look at the kidney blood flow. And you can see here, this has a ultrasound probe over the renal artery, and this is a laser tissue Doppler that sits over the cortex to look at capillary perfusion in mice. And so we give a much, even more doses of angiotensin 2 in this sort, because this is really a fine, delicate system that gets regulated tightly. And we wanted to look at very small doses up to those big doses we saw where blood pressure's on the top. And this is renal blood flow by an ultrasound device. And at the end is epinephrine again. So the first thing we looked at is just baseline renal blood flow. And you can see just at baseline, you sort of have a scenario where you have enhanced renal blood flow in the smooth muscle knockouts, likely because they have lack of that endogenous ability to constrict the afferent arteriole in these animals. So what happens when you give angiotensin 2? And unlike what we saw in the systemic circulation, the kidney really just completely lacked a response to angiotensin 2, where you see the black squares is the controls and 3 different doses of angiotensin 2, whereas the triangles that are open are the smooth muscle knockout mice, and they really don't have much of a response at all. So really the kidney microvasculature is really affected and does not have that sort of compensatory mechanism that systemic circulation has. Our next experiment was to do a sodium balance. Again, what we do is we measure the sodium every single day, and so we know how much they're eating. We measure their food every day. We measure their urine every day and measure how much sodium content. We do that actually for 5 days. In the first 2 days, it's just to get them acclimated. We then implant an osmotic mini pump, 1,000 nanograms per kilogram per minute, as we always do. And then 5 days, we again measure angiotensin 2. And so the idea is to see over this time period, what do we see for the daily sodium balance? And here are the data for this, and you can see this is like in and out. So you kind of like we balanced how much is in and out, and we sort of come up with this baseline, sort of pretty much even. And then right when you give angiotensin 2, the control mice really start to retain sodium. You can see here over all those 5 days. Now, this is a really challenging study to do, and there's a lot of variabilities that, it's like, for instance, you can have food that gets chewed up and you don't account for it. They don't actually eat it, and urine that spills. So you have to do a lot of animals over many days to kind of get the data here. But you see the smooth muscle knockout mouse don't really have that same response. And if you really sum up the data over 5 days, they're pretty much net even, whereas in the controls are net positive. So this sort of data, again, drives home the importance that the vascular system and angiotensin 2 response is really mediated by the kidney and sodium. So to summarize this, deletion of AT1A receptor in vascular smooth muscle leads to reduced blood pressure and exaggerated salt sensitivity, which I showed that data to you. The hypertensive response is attenuated and associated with enhanced sodium excretion. Despite the elimination, there was substantial preservation of Ang2-dependent vasoconstriction in the systemic, but not in the kidney. The next question we had is, we have these profound changes in sodium. What's the cause of that? Is it all just a total effect of more sodium is being dumped into the vasculature, and that means they're losing it into the tubules, and then you're having enhanced sodium excretion? Or is there a crosstalk between the vascular system? So our hypothesis that we'd actually see compensation. So if you look inside the tubules, you'd be dumping more sodium, and they'd actually be turned on to try to grab some of that back. And so that was the hypothesis going into that. Now, these experiments were done in collaboration with Alicia McDonough, really is just a master at assaying all of these different channels. And so we looked at the cortex and medulla, and we basically looked at every single sodium transporter that was involved in regulation of the system. We did this at baseline and with angiotensin too. And so this is sort of just an overview of all the Western blots that were done. And so I'm not going to go into detail on these, but I want to show you sort of what the data looks like. So here are the data, and this is just looking at what is the difference in the smooth muscle knockout mouse normalized to the wild type at baseline, to say like, okay, are these up or down or the same? And remember, our hypothesis is that many of these would actually be upregulated in an effort to compensate for loss of the sodium in the urine from this low blood pressure state that these mice have. However, we found the opposite. NKCC2 in particular was lower at both the cortex and in the medulla. We also saw lower NHE3 in the medulla. And so this is something that we were really shocked to see, that because that would also imply that there was actually a contribution of the tubules to contribute to the lower blood pressure and enhance sodium excretion. So they're actually both working in concert together to reduce the sodium excretion by the kidney. So let's move on and sort of look at this a little closer just to see the data. So this is, I had mentioned, in NKCC2, both in the cortex and the medulla was lower. And that was pretty profound. And we're going to sort of look at that a little bit more. And then NHE3 also was reduced. So is this really a functional issue, or is this just biochemical assay? You know, is there any difference in the action of NKCC2? So to address this, we gave furosemide to our mice, 25 milligrams per kilogram intraperitoneal, and then 4 hours later collected the urine. And we first just gave them a vehicle and really saw no difference in sodium excretion. And remember, these mice are at homeostasis, very similar to our sodium balance study before we gave the medication. So if you recall earlier, what we saw is that NKCC2 was lower in the smooth muscle knockout mice, and we gave furosemide to block it. You see a blunted natriuretic response, and that really shows that, well, this appears to be a functional diminish. So you have less NKCC2 to block, and you have less natriuretic response. So this gives us a little bit even more. It's like there is some connection between the vascular angiotensin receptors and some signal that they're giving to the tubules. So our original hypothesis is that we'd see enhanced sodium channel abundance and function, and we saw the opposite. Thus, changes in the tubular function seem to be a primary disturbance in mediating the sodium excretion and blood pressure effect in this system. What about with Ang2-induced hypertension? And so we used the same model of angiotensin 2 infusion, and we assayed all these same sodium transporters and channels at day 5. We picked day 5 because that's also where we had our sodium balance data, and it's also where we have our biggest change in blood pressure after giving angiotensin 2. And so the very first thing is to look at what happens in the wild-type mouse. And so there's a lot of data looking at infusing angiotensin 2 in wild-type mice and all the alterations that can occur in that scenario. And so I think it's really important just to kind of look at that first, and then we'll look at the smooth muscle knockout mouse. And so a couple of things to note here. One, there is this phenomenon called pressure natriuresis, where when you have an increase in blood pressure, you have downregulation of some of the tubular compartments as a way to bring that blood pressure down and lose sodium. And so that typically is seen with NHE3. And you can see that NHE3 is diminished in these animals as well as in NKCC2. However, we also know that there are many other sodium transporters and channels that actually contribute to that blood pressure elevation, in particular ENaC. And so ENaC gets activated. You have increase in aldosterone. And so you can see you have increase in alpha and gamma activation of ENaC. You also see in the cortex in NCC activation. And so this is sort of a pattern that we see in the wild type scenario. So what happens in the smooth muscle knockout? So this is another very interesting thing here. And it's something we were definitely not expecting at all. And what we see is a lack of the pressure natriuresis response. So NHE3, which typically goes down, is like pretty much unaffected. And we hypothesize potentially that this does not have to turn on because the blood pressure really doesn't go quite as high in these animals. The second thing we found is that ENaC is greatly diminished in our smooth muscle knockout, both in the cortex and the medulla. In fact, it is almost 75% reduced. And this was really shocking to us too, because we had, just a few years prior, knocked out the type 1 angiotensin receptor in the principal cells of the collecting duct. And this vascular knockout had a more profound impact in ENaC than the principal cell. And so that was really an unexpected finding that we were not, that we just weren't expecting to see. And so this is just some of the data. You can see cortical NHE3, you see that down regulation, so that pressure natriuretic response, and really no change in the smooth muscle knockout. And very similar thing that you see in the medullary NHE3. And then here are the data with alpha ENaC, where you see this huge upregulation of cleaved alpha ENaC in the cortex, in the medulla, but you really see a very blunted response in the smooth muscle knockout, and a very similar thing in the gamma ENaC. The next study is just very similar to what we did with the furosemide, as we gave these animals amiloride, but we also timed it up to when they received angiotensin 2. So we gave angiotensin 2, and then a few days later, we gave amiloride as an IV bolus, and they had angiotensin 2 chronically via osmotic mini pump. And then we see how much urinary excretion of both urine volume and sodium excretion. The one thing about amiloride, it's definitely not as potent as furosemide, and you see that you don't have a whole lot of change in urinary volume, but you do have an effect in sodium excretion, whereas in the wild type scenario, you do have activated ENaC, not much at baseline, but with Ang2 you see increase in natriuresis, and this was blunted with the addition of the smooth muscle knockout. Again, showing that this is not just a biochemical change, but actually a functional change, which with less activated ENaC for amiloride to work on. The last but not least is we wanted to make sure there's no alterations in aldosterone. Aldosterone is increased with angiotensin 2 infusion, but there's really no change in aldosterone. Since we saw those big effects in ENaC, we wanted to make sure that wasn't the case. And then lastly, we looked at glomerular filtration rate at baseline and did not see an effect on glomerular filtration. However, we did not look at Ang2 induced hypertension, which could see an effect in that SNATE. So to summarize this data, we have a scenario where this is just at baseline, you have decrease in HE3, you have decrease in KCC2, in both cortical and medullary, and you also have this reduced natriuretic response to furosemide. And if you look at the Ang2 scenario, we have several alterations. One is you have this lack of NHE3 response that you typically see in the baseline state. You see lack of alpha and gamma ENaC in both the cortex and the medulla. And also this reduced natriuretic response to amiloride. Summarize that mice lacking vascular AT1A receptors fail to generate this proximal tubular pressure natriuresis response, which could be just from the lack of hypertension that is seen. And the lack of vascular AT1 receptors leading to reduced NKCC2 abundance along the loop of Henle and at baseline. And you have this improved natriuresis during angio infusion in mice that lack the AT1 receptors with reductions in NKCC2 and diminished ENaC activation with reduced response to amiloride. So our data suggests that there is some vascular epithelial crosstalk that modulates renal sodium handling and thereby contributes to the control of blood pressure at baseline and during hypertension. This crosstalk is an important insight into the physiologic mechanism, the pressure natriuresis response, and the underlying hypertension pathogenesis. With that, I'd like to thank a lot of people that really helped me with this project, most particularly Dr. Kaufman, who is my mentor, and also the rest of the lab, including Susan Gurley, who I mentioned earlier. Steve Crowley did a lot of the studies, and my mentor as well, and everyone else at Duke. I particularly want to thank everyone in my lab, Fitro Rianto, who just completed his postdoc and now is doing a pathology residency in New York, Imre Dilman, who was a visiting scholar from Netherlands, and then a plethora of other students and now med students throughout the country. And also I thank all the funding agencies that help support this work.
Fantastic. Thanks so much, Matt, for your presentation. We're going to jump right into some of the questions that are being asked right now. And just a reminder to the audience once again, if you do have a question, you can submit it using the Ask a Question panel here on your screen. We may not have time to cover everything during the live session, but we'll be releasing a Q&A report in the coming weeks, so stay tuned for that. Okay, Matt, so first question. Why did you mainly use female mice?
Oh, well, I think I used in those experiments I showed you were all male mice. I did show a few slides that had female mice just to show that we have repeated them, but the main reason is they were mainly for historical reasons. Some of the challenges in doing kidney transplantation in female mice were readily known, and so those experiments were done on male mice. And I think also really the push to do mice in both sexes, male and female, really occurred maybe a decade ago, right when sort of these studies were done. So we have done all of these studies on female mice, and they are published. What I did show was male mice.
Okay, great. Another question for you here. Did you do any studies on APOL1 subtypes in combination with the AT1B or A receptors on blood pressure?
Rodents do not have APOL1. Therefore, if you were to do experiments on them, you'd have to have some mice that have been made that have it. So that's been one of the limitations for research in that gene. I think it could be a very interesting study to do. Those mice are now made. Catlin Sustak basically made mice that have APOL1, the human G1 and G2, and G0, the wild type alleles knocked in. So those studies have not been done, but could be an interesting thing to look at.
Absolutely. A clarification question perhaps here. So maybe you could clarify this one. So the effect of angiotensin 2 in elevation of blood pressure is mainly on smooth muscle cell action more than tubular sodium reabsorption?
Yeah, I didn't show the data for the tubular knockout mice, but we see a very interesting pattern. I mentioned some studies from Susan Gurley published in Cell Metabolism that knocks out the AT1 receptor in the proximal tubule. And I think if you look at both the proximal tubule and the smooth muscle, then you almost would have absent response to angiotensin 2. Proximal tubule does have a fairly profound effect on blood pressure elevation. So it's a combination. I'll also say that if you look at other tubular segments, for instance, the intercalated cell of the collecting duct, it actually has a buffering response. So if you infuse angiotensin 2 into an intercalated cell, AT1A knockout mouse, you actually have high blood pressure. And so the thought is that there is this mechanism by which you dampen the blood pressure response from the intercalated cell of the collecting duct, but enhance it in the proximal tubule. So not that's a long answer, but I think it's a concerted effect of both vascular smooth muscle and proximal tubule cells of the kidney.
Perfect. Thanks very much. There's a lot of questions that are coming in here, so I'm going to do my best to try and get through as many as we can in the next 10 or so minutes.
Sure.
So here, the next question we have here, why did blood pressure not increase in the wild-type mice, those who received the high-salt diet?
That's a very curious effect, and so that's been seen in C57 black 6 mice. They are very resistant to blood pressure elevations, even with high salt. Now, for whatever reason, the C57 black 6 mice that's from Germany does not quite have the same effect. So, I don't know if something has happened over time. I think for us, I think it was nice because if you have a 129 mouse, for instance, those are very salt sensitive, and so they'll have huge blood pressure decreases and increases. I always looked at it like the C57 black 6 mouse has more of a vascular phenotype, and the 129 mouse has more of a tubular phenotype in respect to angiotensin actions. But the exact scientific rationale and reasoning for that, I don't think has really been completely explained, but it's an interesting question.
Absolutely. Another one here, does angiotensin 2 play a role in upgrading sympathetic receptors on blood vessels?
That's a good question. I don't know the answer to that. Definitely, it increases norepinephrine content. What it does to the receptors, I'm unclear about. So we did not look at in receptor upregulation, though. Looking at GPCRs is a tricky business. And as we know from the angiotensin world, there's really not any good antibodies to do Western blots. So you can't do Western blots. If you take a angiotensin antibody into a Western blot in a global knockout mouse, you'll get a great signal. Same way with many of the GPCRs, like the adrenergic receptors. And so then you have to rely on mRNA. And then, mRNA is not necessarily reflective of receptors that are made and put on the cell surface. And so I think it's a very challenging area to interrogate, especially in animals. I would not be surprised if there was some synergy between these two systems. I just don't know the answer to that.
Well, while we're talking about the blood vessels and receptors, has your research touched on the role of nitric oxide as a mediator between tubular and vascular crosstalk? And if so, what does it show?
Perfect. All right. Well, that was our first thought. This is a nitric oxide mediated effect. And so if you give angiotensin 2, nitric oxide is small enough to where it can also traverse cell membranes. So you would have alterations in that. And so what we did is we gave L-name to the mice. And the first thing that we saw was that really, if you would think that L-name was mediating some of the blood pressure response, if you gave L-name, you would see the SMKO mice, their blood pressure would go up to the same value as the controls. And you didn't really see that. You see the blood pressure at the bottom is still different, and then it's different after you give L-name. And then we looked at NKCC2, and that was really the biggest effect of tubular change that we saw. And so we did the exact same experiment where we looked at the uninfused. We saw a difference in cases. There's a whole different cohort of mice even. And then if nitric oxide was mediating this effect on NKCC2, you would see this difference go away. And we really did not see that. In fact, it maybe widened more. So that really sort of think, well, maybe nitric oxide is not playing a role. And this was like one of the very first experiments we did. I will say that I spent probably 4 years trying to decipher this system out, and came up with a lot of different potential thoughts about this. And so, is it inflammation? Is it COX-2 metabolites? What is it that might be perturbing the system? So we looked through a lot of these as well. And we unfortunately could not really identify one specific target. And this is just the prostaglandins, so it's really unchanged. Thromboxane, prostaglandin E2, prostaglandin F. That's a really good question, though.
Yeah, it looks like a number of people had the same thought as you, looking at endothelial-derived nitric oxide as a possible way forward there.
Also, one thing that's interesting is the endothelial. So there's a group that did angiotensin or AT1A receptor upregulation in endothelial cells of mice, and those mice actually have lower blood pressure. And so that's really interesting. It's nitric oxide mediated.
So here's another question for you. Is there any role for the sodium-potassium ATPase pump?
I feel like I'm just setting this up. So we did some RNA sequencing, and here's my next slide. And this was really fascinating as well, because we had several different targets to go after. Now, RNA sequencing is tough because you see differences, but you're unclear the physiological significance. Could it just be a blip? And so we went after some of these blips. And so you see some of the interesting one. One is TMPRSS4, which is involved in ENaC processing. Interestingly enough, TMPRSS4 is actually altered in the opposite direction than we would anticipate. FixIt6 became a target we became very focused on because FixIt6 can actually sort of become part of the sodium-potassium ATPase. And so we really started focusing on FixIt6. I'm not sure if I have the data for that here. Here we go, FixIt6. Yeah, functions in altering the kinetic activity of the sodium-potassium ATPase. So it almost is like sort of a cog in the wheel, if you'd say. And so that was something where like, wow, is this actually just globally altering the ability of the of sodium-potassium ATPase to work? And so we really looked hard. It is just very low expression and really hard to localize where this was. And so we ended up sort of going down a rabbit hunt and really could not decipher if FixIt6 really had anything to do with it. The other thing I'll say is all this is with baseline baseline samples and not with angiotensin 2. So, that's a really good. And you also can see that these mice, the knock-in for SM22 is called taglin, and you can see that the taglin is lower in these mice, again, showing that the knockout was really working.
Well, fantastic. I think I'm going to bring this session to a close. So thank you once again, Matt, for your insights, your presentation, and the answers to all those great questions today. It was really great having you.
Oh, enjoyed it. Thanks a lot for coming, and thanks for all the questions and interest.
A big thanks to you, the audience, for participating. And last but not least, we'd like to thank our sponsor, Alzet Osmotic Pumps. In closing, we hope you enjoyed this Inside Scientific webinar produced in partnership with the American Physiological Society. And we'll see you again next time.